So, dear students, in our last lecture, we have discussed about antigens, how the antigens, formed, what are the types of antigens, how they behave, and how our body's immune system treat the foreign particles in the form of antigen. Today, I will discuss a new topic, that is adjuvants. So, what are adjuvants? Adjuvants are some immunological agents that are also considered as pharmacological agent. And that particular agent has the capability to modify the effect of the other agents. What are the other agents? Other agents is basically the antigen. So it has the ability to modify the effect of the other agent. And substances of this type that can be regarded as adjuvant, when mixed with an antigen and injected into the host parenterally, is parenteral administration, it will enhance the immunogenicity of that particular antigen. So mostly in case of vaccine administration or vaccine treatment, these adjuvants are used to boost the immunogenicity of the injected antigen. This particular word was derived from a Latin word, and the word was adjuvare. Adjuvare, its English meaning is to help, to assist. So these molecules are actually assisting the antigens to boost up their immunogenicity into the host. So that is why they are called adjuvants. So actually what they are doing, they are boosting the immunogenicity of the antigen. Side by side, they also boost the immune response. Means when an antigen has low immunogenicity, or when only small amount of antigen is available, then that particular antigen should be mixed with an adjuvant, and now that particular antigen will strongly show immunogenicity as well as immune responses. So as I said, mostly they are added to a vaccine to modify the immune response. So how they can be added, how they can modify the immune response? So actually the motto or the goal of vaccination is to boost up your adaptive immunity, to initiate or activate your adaptive immunity, because your innate immunity is not sufficient to combat with different type of diseases, different type of pathogens. So we have to boost up our immunity. We need an additional protection system, and this particular additional protection system is the vaccination. So excuse me. So here, in the case of vaccination, we are adding adjuvant along with the antigen that is called the vaccine. It may be a toxin, mostly in their toxoid form. It may be a live or attenuated vaccine. So you have already studied what are the types of vaccine. So mostly this particular process is used to produce more antibodies to induce the humoral immune system as well as the cell-mediated immune system, so that both the B type of cell and T type of cells, and B cells and B lymphocytes and T lymphocytes, they induce the production of more amount of antibody in our body. And the second thing is that they can last in our body for a longer period. It's not that the person or the individual has been administered a vaccine and within 3-4 months he or she will get infected by a particular disease. For that, we have already used vaccination to combat against the disease. But being with this protection system, still we are getting infection. So the point is that we are doing vaccination to immune the individual, and that individual should be in the state of immune that he or she can combat against the particular disease for what reason vaccination has been done. So he or she can fight against that particular disease for a longer period. So in a sustained way, in a long-lasting immunity, to provide a long-lasting immunity, these adjuvants are used to boost up the immunogenicity of the antigen. This we can, so if we can get success for a long-lasting immunity, so then definitely we can minimize the dose of antigen use. We can minimize the cost, definitely cost of vaccination, because if periodically we have to get vaccine, then not only we are injecting the foreign particle in our body for multiple times, but also it is the cost, because in few cases to prepare the vaccines they are very much costly. So not all the people can afford this. So minimizing the dose as well as the cost of vaccination can be reduced. So these are the commonly used adjuvants. Mostly the aluminum compound, they are mostly used as an adjuvant. And the first aluminum salt used as an adjuvant is aluminum potassium sulfate. That is also known as alum. So now completely replaced by aluminum hydroxide and aluminum phosphate for the production of commercial vaccine. There are some other vaccines, other adjuvants are also there for the production of vaccine. The list is here. You can see that Triumph's incomplete adjuvant, Triumph's complete adjuvant, mostly oil in water emulsion, and then Mycobacterium tuberculosis in its inactivated and dried form can also be used as an adjuvant. Then plant saponins, cytokines, detergents like QLA, mineral oil or food-based oil such as peanut oil can also be used. Toxoids and lipopolysaccharide derived from gram-negative bacteria, then killed Bordetella pertussis, that is the causative agent of pertussis, and also some synthetic polynucleotide can be used as an adjuvant, and they can be used for the preparation of mostly vaccine. Now, how they are functioning, what is the mechanism behind this immune stimulation by the adjuvant? So these are four different type of mechanism. One is to extend the presence of antigen in the bloodstream, that is for sustained release, sustainability of the antigen into the bloodstream of the host. Second one is by boosting or helping the antigen presenting cells to absorb the antigen. And third one is to activate the macrophages as well as the phagocytic cells, lymphocyte cells. And third one is for boosting the production of the cytokines. So here, with this figure, you can get some idea. So here you can see how the adjuvant, I have taken an example of alum adjuvant, because mostly the alum or aluminum based nano adjuvants are used nowadays for the production of vaccine and to boost up or induce the immune responses. So recently, it has been shown that the aluminium adjuvant could activate the nucleotide bindings of nucleotide binding and oligomerization in leucine-rich repeats pyrin domain here this NLRP3. That means that nucleotide binding leucine rich repeat. These are LR leucine rich repeat of pyrin domain containing 3. That is, the short form is NLRP3. So actually this inflammasome and uh, the other thing is interleukin 1 beta, this red cell, this red one. So this is the inflammasome, and here is this interleukin 1 beta. So inflammasome and interleukin beta production, they are activating this. And then which could explain its ability to induce the local inflammation. Now you can see local inflammation here. That is why redness after injecting the vaccine, you can see the redness of the local area. So that is only because of the local inflammation induced by this NLRP3 inflammasome activation as well as interleukin 1 beta activation. Now, what will they do? They have the ability to induce local inflammation as well as they will now recruit the antigen presenting cells, the APCs. Here you can see the antigen presenting cells to enhance antigen uptake, because that is one of the reasons. Antigen uptake first, here you can see antigen uptake. So they will induce the antigen presenting cells to take up or uptake the antigens, and also inducing the dendritic cell maturation. Here you can see DC maturation, this dendritic cell maturation. And this IL-1 beta will stimulate the T cell, T lymphocytes. So they will activate and stimulate the T lymphocyte, and then the T lymphocyte will induce the B lymphocyte or the B cells, and these B cells will then synthesize or produce the antibodies. So that is how the administration of a particular antigen in the form of vaccine. So antigens are mixed. Here is the antigen. They are mixed with this adjuvant alarm, and then they are administered intramuscularly mostly. So they will report effects like that. They will just go for this dendritic cell membrane perturbation. Then they will induce the binding of nucleotide binding of and oligomerization of leucine-rich repeats, pyrin domain containing 3 inflammasome activation. Here you can see they're coming here and inducing this inflammasome. Now they are activating so many things. They're activating the CDs, they're activating the mesohistocompatibility complex, and this can be received by the T cells with their receptor, T cell receptor, you can see. And also they will activate interleukin 1 beta. Interleukin 1 beta will induce or activate the T cells. Now they will induce the antigen presenting cells to active or receive more antigens. They will also induce the dendritic cell and mature them, and then they will induce the B lymphocytes for the production of antibody. This is the mechanism of adjuvant, how they are boosting the or inducing the immune response in our body. Now, a question will definitely come into our mind that what is the utility of adjuvant? Why can't the antigen only can do all the functions? What I have discussed in my last slide. So this is a comparison between administration of an antigen in the form of vaccine solely and mixed the antigen with an adjuvant and then injected in a form of vaccine. So what is the comparison between the two? What are the effects they will do? They will create in our body. So mostly after injecting the antigen in our body in any of the forms, without an adjuvant or with adjuvant, it is the macrophages. They will come, they will interact with macrophages, then dendritic cells, and these dendritic cells will be the immature dendritic cells. Immature dendritic cell, mature macrophages, monocytes, they will come and interact. And the first thing they do, they will just induce some pro-inflammatory cytokines as well as chemokines. And these pro-inflammatory cytokines and chemokines, they will induce the antigen-presenting cell to take off, to uptake the antigens. Here you can see the antigens present there. So here is the antigen. So they will induce to take off the antigen. Here, this square particle, you can see. So these are the antigen. So these antigens will be taken off by the antigen presenting cell. So this should be the mature antigen presenting cell. And now they will induce cytokine release. So these cytokines will activate the CD4 active T cell. They releases the cytokines, the CD4 active T cells. They release the cytokines, and these cytokines will induce the B cell, B lymphocyte. Now the B lymphocyte will have dual function. The plasma cells, they will produce antibody, and the memory B cell, they will memorize this function. So here you can see that the plasma cells, they are actually synthesizing antibody after activation of the B lymphocyte by the cytokines released by the T cells, and also they will memorize the function. But now here you can check. So this pentagon, this green color pentagon, these are the adjuvant, and this is the antigens. Now the antigen is mixed with the adjuvant and then administered in the form of a vaccine. So same thing, the innate immune response, innate immunity. So at the site of injection, the macrophages, the immature dendritic cell, the monocytes, they will come and they will create the similar symptoms, similar condition here, means release of pro-inflammatory cytokines as well as the chemokines. And now these will activate the antigen presenting cell, and the antigen presenting cell will activate the T cells, mostly the CD4 class T cells. So here you can see that the cytokines will be an improved condition cytokines. Wow! Because here you can see that the CD4 T cells, now they will release a different type of cytokine. Here by showing different color, we are just trying to discriminate these two. So here you can see different type of cytokines will come and induce B lymphocytes. Now B lymphocytes has three functions. First, the plasma cell, they will produce antibody. The memory B cells, they will memorize all the things. And the third one is that the second type of plasma, the plasma cells here, they will produce a huge number of antibodies. So here production of antibodies means induction of adaptive responses by the effector cells. So what are the effector cells? The effector cells are these are relatively short-lived activated cells. And what is the function of the effector cells? What are the type of the effector cells? Now, these are the short-lived activated cells that define the body immune response. How? Mostly the effector B cells, they are called plasma cells. Here you can see these are the effector cells, short-lived cells, plasma cells. So they secrete antibodies and also activate the T cells, include cytotoxic T cells and helper T cells, which carry out cell-mediated immune response. So they induce two types of T cells, cytotoxic T cells and T helper cells. And also they are inducing the plasma cells to produce more and more number of antibodies. So through this picture, you can see that here the antibody production is less than that here, only because of the presence of the effector cells. The effector cells is not long-lasting, it's very short-lived. They will induce these cells, T helper cells, cytotoxic T cells, and the B cells, mostly the plasma cells, induce production of antibody, huge amount of antibody will come and mix with the blood, and then they will be eradicated. So they are actually boosting the body immune response, boosting the body's adaptive immune response. So that is why nowadays adjuvant are used along with the antigen, and we have discussed what are the types of adjuvants can be used. So if we can summarize, so adjuvants can act like a pathogen-associated molecular patterns that triggers the pathogen recognition receptors, PRRs, and they will just act like a pathogen-associated molecular pattern. So our PRR will identify this adjuvant as a pathogen and will trigger the immune response, innate immune response basically, through a variety of mechanisms. We have discussed the four already, to identify the vaccine components as a threat. This is important. The vaccine components should be treated as a threat. Then only the innate immune cells responsible for innate immunity will come and play and will induce the T cells as well as the B cells for the production of antibody. So this should consider, this should identify, this should recognize adjuvant as a whole, the vaccine as a threat to our body, and then activate and mature the antigen presenting cells. Now the antigen presenting cells will uptake both the adjuvant and the antigen and will initiate the downstream adaptive immune response. So this is how the adjuvant are functioning and boosting our innate immunity. That's all.